would help if the microphone was on. So sorry if anyone at the back couldn't hear me before that, and sorry to everyone online who probably couldn't hear me at all then. So yes, great time to be someone that likes data. We've got lots of data we can collect, we can handle and analyze these data. So today I'm just going to be giving you a whistle-stop tour of how we try to capitalize on this and some of our plans for future work. So a few years ago, we really wanted to dive into the literature on refinements to laboratory rat housing and husbandry so that we could see what had been studied and how it had been studied to try and draw some conclusions and make well-informed decisions about how we should be caring for our rats. And we didn't have to go to the library and trawl through lots of printed journal articles like we'd have had to do, a few decades ago. We could just use online databases like Web of Science to find relevant research articles. And then although we had a lot of hits, we could use software like Rayon to really efficiently screen through all of those abstracts. Then once we had all of our full text articles, we could use file hosting services like OneDrive to work together as a team to extract all that data. All of this meant that it was feasible for us to conduct a mapping review of over 1,000 articles detailing potential refinements to laboratory rat housing and husbandry. This is now published, and this was funded by UFAW, so thank you UFAW for funding this work. We've published a database of relevant articles alongside this that you can go and search by topic. We've identified various knowledge gaps, so areas where research would be really, really valuable. And we've also highlighted the importance of appreciating individual differences when thinking about implementing these refinements and thinking about how to study further refinements. We've also conducted surveys of pet rat owners to better understand rat welfare, and surveys are a really low-cost way of obtaining large amounts of data that really lend themselves to exploratory research. We've used social media to reach as many potential owners as possible, and that's really helped to get much larger data sets. And we've also used R to clean and to analyze those bigger and actually really, really messy data sets. We've published 3 articles based on the results of those surveys. In one, we've highlighted some welfare concerns, including that many pet rats are exposed to predator species, so species like cats and snakes. We've suggested that digging and bounding and bruxing might be useful as welfare indicators because we see that the frequency at which these are observed depends on the level of exposure of these rats to the predator species. We've described this really cool and fun behavior called boggling in rats, and you can see this little guy doing that here. And this is something that had received very little or not any scientific attention, but something that pet rat owners report seeing fairly frequently. And we've also used factor analyses to assess whether these surveys might be useful or provide useful tool for studying stable individual differences in rats, so what we think of as personality. And it seems like this approach might be quite useful. We've also conducted operant studies in the lab with rats, and we've automated this process so that we can just take a rat, put a rat in the box, wait outside for half an hour watching the rat on CCTV, come back in, get the rat out of the box, get the data off of the computer. And this automation has a couple of benefits. It first of all means that we can train and test multiple rats at the same time for really high throughput data. We can collect lots of data. But it also means that we reduce the potential for the risk of biases because experiments are just less involved in the data collection. Now, there are lots of studies that you can run with rats in operant box. We have just published, literally in the last 2 weeks, details of a novel study. And this is a really, really simple task. It just took us 6 training sessions before rats were ready for testing. You might be able to reduce that. And these rats are not food or water restricted at all. So in this task, rats are able to visit 2 separate food troughs on opposite sides of the box to get some food. So they just stick their head in a trough, they wait for an amount of time, and then some food arrives. If they make the state response, so if they stay at the same trough, then we increase the time they need to keep their head in the trough before we'll give them some food. If they make the switch response, so they go to the other side of the box, then the timer's just reset. So rats are just having to make decisions about whether to stay or whether to switch. And rats really are making a lot of these decisions. So they're making well over 100 decisions on average in a 30-minute test session. So we're running this test multiple days per week over multiple weeks. You can see that we're getting a lot of data. And the idea is that decision making on this task requires rats to have various expectations. So expectations about how rewarding their current location is, expectations about how costly it is to get across the other side of the box, and that these expectations are things that should be informed by emotion and so welfare. So that we then tested this by testing rats in different conditions, under nice conditions where they're in spacious complex housing, lots of different bits of enrichment. Then in a period where we took away some, but not all of that enrichment, or we put them through unpredictable housing. And as you can see from this plot, when rats were in those periods that we think should be associated with poorer welfare, they were much more likely to make that switch decision. And what I really like about decision-making tasks is that we can often use computational modeling to better understand the data and to better understand that relationship between emotion and decision-making. And really, really broadly, computational modeling is a formal and a mathematical description of the processes that generated the data. So they really allow us to get into the nitty-gritty of that relationship between decision making and emotion. And I think computational modeling is really useful for studying emotion because emotion is something that we can never directly observe. We have these various different measures of emotion, but they're not allowing us to directly observe the emotion because they are observing the consequences of the emotion on something like behavior or physiology. And although computational modeling doesn't allow us to directly observe the emotion, it does allow us to break our measure down into the various processes and look at how they might be influenced by emotion. So in the example of decision making, breaking that decision down into its various subcomponents, so things like how hungry the animal is, how tasty the food is to the animal, how quickly they're able to run, what they think the probability is that there's a predator in the box, and then asking if and how emotion might be influencing each of those subcomponents. Now, this combination of running decision-making tasks and then using computational approaches to analyze the data has been used really, really successfully to better understand affective disorders, so things like depression or anxiety in humans, to the extent that it's a whole field now. There is a whole field of computational psychiatry. There are multiple textbooks, there's an annual conference, there's a journal, and I think this approach could be really useful for us to use to study emotions in animals too. So many of the studies in computational psychiatry use a reinforcement learning framework for building those models. And reinforcement learning really emerged from animal behavior studies, really early animal behavior studies. So I think that it's going to be a really useful approach to us use too. It's going to be easy to translate. And really just in a nutshell, reinforcement learning is about how an animal learns about their environment through their interactions with the environment to maximize the rewards they get and to minimize their losses or the punishes. So for example, if there's a rat just living in the world, how this rat learns to go towards the trees where all the snails are hanging out to have some food and avoid the trees where there's a bird going to eat them. And this is clearly not to scale at all. So a really, really important and a really interesting subcomponent of that full reinforcement learning problem is classical conditioning, which I know many of you are going to be familiar with, but I'm going to run through it with a knee-jerk example. So in this example, the human, other animals show a knee-jerk reflex. Cats, I think, do. So you can imagine cat instead, but put a human up. The human is just quite passive in their environment. A bell rings, a hammer hits their knee, that elicits the knee-jerk response. Now, at first, the bell has no meaning to the human, but through repeated pairings between the bell and the hammer, the bell comes to elicit that knee-jerk response on its own without the hammer being anywhere near the knee. This can be modeled really simply and really easily using this beautiful learning rule, this model of learning that was first described by Bush and Micella, and then was later extended by Rascola and Wagner. And the rule just states that the updated predicted value, so the expectation when the bell rings, is equal to its current value, added to the difference between the outcome and the current value, where that difference is scaled by a learning rate. And the learning rate is just a value between 0 and 1 that characterizes how quickly learning takes place. This difference is sometimes called the prediction error. It's the difference between what we think is going to happen and what actually happens. So really what it reflects is surprise. How surprising is that outcome? So the learning rule just states that learning is all about surprise. Now we can think about, or use this rule to think about, how an animal's behavior might change when when they have different learning rates and when we provide them with different levels of reward. So going back to thinking about a rat in a box, just imagine a rat in a box and they're going to the trough and we're giving them different amounts of sucrose. And we decide that we're going to vary the sucrose systematically so that it just goes up and down and up and down. So all of the crosses that you can see on the plot represent those hypothetical rewards that we're giving to the rat. And then we can think about what is the value of approaching the trough of the rat if they had a different learning rate. So you can see that if a rat had a learning rate of 0, then their value of approaching the trough would always be 0. They're just not learning anything about what's happening in the trough. But as the learning rate increases, the rat's value of approaching the trough more closely tracks the actual volume of sucrose that they're getting. So these rats with high learning rates are always just less surprised about what's happening in their environment. And you can see that in the subplot too. So as the learning rate increases, the average squared prediction error decreases. Those rats with high learning rates are less surprised. But what if we change the structure of the rewards? So what if this time we keep the rewards pretty much the same, and then we just throw in some really high value rewards? So you can see that now all the crosses on the plot. Well, if a rat wanted to reduce how surprised they were in their world, how well they knew their environment and responded in an optimal way for their environment, you can see that actually having the learning rate, the highest learning rate, wouldn't be sensible for the rat, because rats, or hypothetical rats with those high learning rates, start to chase those high rewards that don't represent what is happening in their environment on average. So we can do a couple of things with this. We can first of all use this to make predictions about what we think should happen to learning when we have animals in different environments that may have different implications for their welfare. So for example, stable versus volatile environments. But we can then also start to test those predictions by going out and collecting data with real rats and fitting that data back to the model. And that's the point at which you need to start thinking more about how those values turn into behavior, and starting building upon that learning rule model a little bit more. And what I think is really, really cool about this is that there is this nice and neat equation for learning that actually has some bearing on the neurobiology of learning. So we think that dopamine neurons in the ventral tegmental area, the VTA, encode these reward prediction errors. So they change depending on whether there's a positive or a negative prediction error or no change in prediction error. And what's more is that we know from human studies that positive reward prediction errors lead to happiness in humans. So humans report feeling happier when things are better than expected. We've also shown this in a task using food rewards with humans, so a task that much more closely parallels the sorts of studies that we use in animals. So you can see that even though initially these computation models might seem a bit cold, they might be really crucial in understanding those links between emotion and decision making, even at a neurobiological level. And now, since starting working with rats, since starting my PhD and running operant tasks, a real bugbear for mine has been the equipment. The equipment is just so expensive. It's really inflexible. I can never do exactly what I want to do. And when it breaks, which it has done on several occasions, it takes a few months for new equipment to arrive. It's an absolute pain. So when I started my fellowship, I thought, why don't I just build this myself? Like Raspberry Pis are really widely available now. There are lots of resources on how to use this. I can code. I can do this. So that's what I tried to do. I bought a Raspberry Pi. I bought some sensors. I bought some motors. I bought a screen. I did some coding. I also enlisted the help of the workshop at Bristol to build me a nice Perspex box, because like laser cutting is just not within my skill set. And they thankfully built me a really beautiful box. And I think this has a number of benefits. It, first of all, is actually really cheap. It costs so much less than the off-the-shelf equipment. It's really, really flexible. If I want to run a different task, if I want to change the task, I just edit my code. I just move some of these sensors about. But maybe most importantly, and linking to the lovely talk that Anna gave earlier, it's really great for open science, because if you wanted to try and repeat these experiments, I could just send you my code. I could just send you a shopping list of all the things that I've bought. You could do it fairly cheaply yourself. I could share all of this with you. So I think it's going to be really great for reproducible research. We're still in the piloting phase of this at the moment. You can see our prototype equipment. You can see one of our pilot rats, but it's looking good. So really watch this space. I'm going to wrap up there. I'm just going to take some extra time to say thank you to lots of people who are willing to thank. So I want to thank everyone in the animal welfare and behaviour group. at the University of Bristol. You can see most of us there. My colleague Jo, who's giving a talk later, sadly wasn't here, but she's going to give an amazing talk. She's an amazing person. In particular, I would like to say thank you to Molly and Nick, who are both here, both colleagues of mine, both working on computational stuff. I can see Nick, I can see Molly. That they've been incredibly, lovely people to work with. I like it when they send me nice plots and nice data. Thank you, guys. And there are 4 people who I really need to thank and say an absolute huge thank you to. That's Mike Mendel, Liz Paul, Peter Dine, and Ian Gilchrist. They were my 4 PhD supervisors. I was very lucky. I got 4 supervisors. They are all just incredibly supportive, wonderful human beings. They're also amazing scientists. You'll know that if you've ever seen Mike give a talk, or if you've ever spoken to Mike. Mike is sitting there. Thank you very much, Mike. I really appreciate it, and I really appreciate having the opportunity to work with them. Of course, thank you to funders, BBUSC and UFAW, and again, thank you UFAW for this amazing award. It means so much to me. Thank you everyone for listening. Happy to take any questions and talk to you over lunch or later. Thank you everyone.
This is more a +1 to your final comment, Vikki. I, in my PhD, built all of my own equipment, and everybody thought I was crazy. I've built my own equipment my whole career. I'm so glad that there's somebody else doing it.
It's so fun.
It is. It's awesome. And I really wanted to +1 as well, the idea of open sourcing this stuff. So right now, all of our projects, like our CAD files, our 3D printing files, our laser cutting files, our circuit diagrams, so I think 10 years ago, there was a culture of patent it and sell it. And instead we're there's a really important concept called theft, where you can release these documents. Anybody can take them, anybody can use them, anybody can manipulate them, but if they start trying to make money off them, then all of that license refers back to the original person. And so again, when we were talking about people being worried about their ideas being nicked, if you're worried about your designs being nicked, copyleft licenses are a really powerful tool.
Yeah.
But I just wanted to say, I'm so excited to see somebody else doing this.
Thank you. And yet I hadn't heard of that. So thank you very much. That's going to be really useful. And it is really fun, and it's so flexible. So if you're interested in building your own equipment, also come and talk to me or Jay. Okay, so next up we have Karolína Steinerová.
Hello, everyone. I'm very excited to talk about part of my PhD that is mostly about play behavior in pigs. And this study has focused on rearing pigs with play opportunities and their effects on disease resilience in pigs experimentally inoculated with porcine reproductive and respiratory virus. And just to start, I want to say a brief definition of disease resilience. Disease animal that is resilient comes back to the pre-challenge status faster. So that's what we were measuring. If the pigs inoculated with this virus get better faster compared to control. So I will be talking about interplay with positive emotions and health, because there is a lot of evidence that in humans, if we feel good, there's some correlation with better health. And there is emerging evidence that this is the same for farm animals and other mammals. So we are focusing on pigs and how can we facilitate positive emotion in pigs? So it's been proposed that the pigs feel positive emotions if we provide them a stimulus-rich environment and enable them to perform rewarding activity. And what is the rewarding activity? It can be a positive human contact with the stakeholder, or it can be offering some biologically relevant enrichment. And there is evidence from studies that pigs that were reared in environment in intensive farming that was promoting positive experiences, they were more resilient against common stressful challenges encountered in the modern production. And we saw better response to disease, specifically to the PRRS virus that I used in my study, also to short-term transport, and also to injury. They healed faster. So in my first study, we wanted to prove that pigs play even when they are older. So we promoted play in older pigs, in grow-finished pigs, and we measured if it's positive for them. So our data indicate that play is positive for the pigs. So we took this knowledge and used it in our next experiment, where we explored whether pigs reared since birth with regular play opportunities develop greater resilience when we inoculate them with the virus compared to control pigs. And we use this virus because it's very industry-relevant disease. It affects their growth, of course, welfare, respiration, and it causes mortalities in all ages. So for our methods, we had our pigs born and we divided them into play and control treatment. And we promoted play in our play treatment from 5 days of age. And we gave them, as you can see on the upper picture, extra space in the farrowing room with different destructible objects that they enjoy, as we have indications from the first study, and with aspects of novelty, because pigs really enjoy novelty as well. And we had our control treatment that was reared in standard farrowing pen, which was smaller. When the pigs were 15 days of age, we performed back tests that was shown to influence the immune response, and we divided them into high, medium, and low resistors. So basically how the pig is behaving when you flip it on the back, whether it's a high resisting or low resisting pigs, you can indicate the coping style. And when the pigs were about 25 days of age, we weaned them and created new pen groups for the disease challenge. So we had 4 pen groups, 2 pens per treatment with 7 pigs per pen. And then we transported them to the disease containment facility, where we actually inoculated them with the virus, and they were kept in the same pen groups created at weaning. And we inoculated them with the control dose of the virus when they were 43 days of age. We collected a lot of measures. We collected behavior, skin lesions. We measured viral load and cell counts. We observed clinical signs relevant to the virus. Also measured some performance indicators as average daily gain and pen level feed intake, and we observed also play behavior and measured rectal temperature. And we euthanized the pigs 22 days after the infection. So moving to our results, first, the skin lesions that we used as a measure of aggression. We see that here on the graphs, you can see blue, it's control, and red, it's play treatment. It will be the same on all of the graphs. And we don't see any differences before and after weaning, but when you look what happened after transport, we have more skin lesions in control, and this was continuing until after inoculation and until the end of the study. So we have some indications that the pigs coped better with the stressful challenge, which is transport is generally stressful for the pigs. For the viral load, it was surprising for us that we didn't have any treatment differences between play and control pigs. We saw a very typical response for after inoculation when the virus is replicating. So there is increase and then it gradually decreased after the 3 weeks, but there were no differences. And for our clinical signs, we observed respiratory distress, and I will be talking about moderate and severe respiratory distress, and we classified it as subtle or obvious abdominal effort with or without increased respiration, which is more than 40 breaths per minute. And if we look first on our number of pigs experiencing this moderate and severe respiratory distress, we have 11 out of 14 play pigs, and all of the control pigs experience this type of distress for at least 1 day. Our predicted number of days experiencing respiratory distress was lower in play pigs. They were suffering from this distress 4 days compared to almost 12 days in control, which was significant. And we also looked what's the probability to experience this distress for per 1 pig. And again, if you are a play pig, you had 35% probability to experience this moderate and respiratory distress compared to 91% in control. So regarding performance, which is probably the most translatable result, we saw that there were differences in higher beneficial for the play treatment. They were growing higher since the beginning, and the difference was more profound in the second half of the infection, from day 13 until day 16 post-inoculation, where they grew about 160 grams per pig more. And this is comparable to healthy pigs' growth for their age, whereas the control pigs were growing less. And I have also some descriptive data for pen level gain to feed ratio, where the play pigs grew, play pigs were more feed efficient. They grew more, 0.72 kilograms per 1 kilogram of feed compared to 0.57 kilograms per 1 kilogram of feed in control. And now we will be moving to our blood immune cells. We measured monocytes, which is very important immune cell for the PRRS, because PRRS replicates in macrophages and monocytes are progenitors of macrophages. And we saw treatment and time effect. So again, PRRS depletes the cells right after inoculation, which we saw in both treatments, but then there were differences occurring where the control pigs were having more monocytes compared to play pigs, and also in control pigs, the monocyte count exceeded the baseline, whereas in play pigs, it was about similar levels. So this indicates that the play pigs might have been experiencing lower inflammatory response. Regarding other immune cells, we found interesting interactions with treatment and average daily gain pre-inoculation, so how the pigs grew from birth until they were inoculated. And in white blood cells and neutrophils, there is the same relationship. We only see differences in slow-growing pigs between control and play. And it's interesting because there is no time effect. This difference was observed before inoculation and during the infection in the slow-growing pigs. And control pigs, slow-growing, had more white blood cells and more neutrophils compared to slow-growing play pigs. And another interesting interaction was with backtest in the measure of lymphocytes. We saw differences in the medium resisting pigs. So again, control pigs, medium resistors, they had more lymphocytes compared to medium resistors play pigs. And this was again irrespective of the stage of the infection. We saw similar effect in the rectal temperature, where there was, on the contrary, difference in high resisting pigs, and high resisting pigs control had the highest temperature, rectal temperature, again, before and after inoculation. So this was not caused by the virus, but this was a general response. And interestingly, we have evidence that the pigs continued to engage in locomotor, social, and object play also after inoculation. So, for example, in the locomotory play, where the pigs spend the most energy, is the fastest and high demanding type of play. We have the highest time of play before inoculation, and then after it decreases, but we see that the pigs roughly played about a minute in the initial of 10 minutes during the play session. So these graphs are only showing first 10 minutes of the play session, where the play is the most intensive. And social play was occurring regardless in very similar time before and after infection. And object play was actually a little bit higher in the second half of the infection. So what is the interpretation of this data? We generated a lot of results, and some results are really hard to interpret. For example, the treatment and backtest interaction, it's hard to conclude something that we can use, but I think the overall message is that the play pigs were less affected by the virus. And they showed, we can say this because they performed better, they grew more, and they were also more feed efficient because of the immune cells results. We proposed that they had lower inflammatory response, but I must tell you that we attempted to measure cytokines, but which are indicators of inflammation, but because of the assays, we were not able to detect the cytokines. So we can only assume lower inflammatory response. And if you remember, the rectal temperature and also the immune cells were showing different interactions, but overall the play pigs created a more uniform group. And it has been proposed that it can be beneficial for the disease resilience for the medical treatments when you have a more uniform group of animals. And they also showed less sickness behavior, as you saw in the respiratory distress. So we can say as our conclusion that there are indications that play pigs that were reared with play opportunities developed better disease resilience. And the implications are improved pig health and welfare. If we use this approach in the intensive farming, we can have more resilient animals. More resilient pigs have advantages against common husbandry procedures such as transport or mixing. And this is the world first evidence that play has effect, or it's suggesting that play has effect on disease resilience, and this opens the door for further research. And this is also interesting because we can now look at play as a fitness indicator, as you saw on the graphs, that the pigs actually respond based on their recovery status to the infection, so there can be more studies done on play as a fitness indicator. Of course, there are limitations. For example, the promotion of play is quite time-consuming and creates extra labor, which is, of course, not wanted in the industry, in the intensive farming. So we are working with farmers where this approach can be used in a more practical way. Also, we have indications about, our study is about play, but can we really say that the effects of on the disease resilience were really attributed only to play? Because there was also more exercise in the play pigs, and we can also say that there was more enrichment given to the play pigs, and this can also have influence on the results. And as I mentioned about the cytokines, we were unable to measure them, so we proposed development of better measures to clarify the unknowns, and also probably we should move to collect some brain tissue to look at dopamine and endogenous opioids that are connected to reward and also motivation. So with this, I would like to thank my research team from the Western College of Veterinary Medicine and also Prairie Swine Center, where we have our pigs, and WIDO for the immune cell measures. And of course, I would really like to thank UFAW that funded me to attend the conference. So thank you very much. And just to finish, this is our website. If you're interested what we are doing, you can see everything on swinewelfare.ca. And now I will take any question. Thank you.
Yeah, thank you for this interesting talk. So, Michael Beaulieu from Wild Animal Initiative. I completely agree with one of the last points you rose, how to dissociate physical activity from play or play from physical activity. But apparently you distinguish different types of play. So locomotor activity, social play, and I don't remember the third category. Have you tried to see to examine the isolated effects of each of these play types on the resilience of the pigs?
Mm-hmm. No, we did not do this in the study. We observed all types of play and didn't differentiate between different types of play. But definitely, I can tell that even though I didn't observe it, I can tell that there might be different effects on the disease resilience. For example, compared to locomotory play and object play, it kind of also we are missing a lot of measures that we can collect. For example, as I mentioned, the brain, if we can measure dopamine, and there are some studies in animals, but we can really see, for example, what type of play is most rewarding. Is this locomotory play or social play? And the object play is very much connected to exploration. So there is a lot of this influence, which is, again, probably more this can be more attributed to the overall enrichment because the pigs perform like play for, let's say, 3 seconds. You see like this excited movement that you can classify as play in object play, but then it continues interacting with the object. So it's like a very long time. So you can't really tell the difference now, but it's very interesting proposal for the next studies.
Mandeep Singh Azad from India. So first of all, we should stop working and play more. It would be good for our health also. Playing is good. Just a suggestion, I think. The sample size you took, don't you think it's a bit small to make sure that this is the only reason that could be the better immunity? It's a nice work, but I think it should be a bigger sample to be sure for this, that this could be the reason. As we can see, same is the case with poultry. If you keep them out, they are more resistant to any disease as when you kept them in a room. So I think, but sample size could be better.
Yes.
So we can authenticate it. It's a nice.
Yeah, I agree. We used 28 pigs, so 14 pigs per group. This was used in another study as well, so where they also saw some differences. So we took it as before we spend a lot more money, we want to see if there are any effects on the disease resilience. So we showed, yes, there are some effects, and the next step is to look in more details and probably more specific measures with a bigger sample size. Yeah, so I agree, the sample size can be higher.
And also next time, try to compare the results with the pigs which are already kept in open, free. Is that the only reason, or special play, or we just keep them in open? Yeah, better.
To differentiate the effects,
yeah,
yes.
it should be.
Mm-hmm.
Thank you.
Good point. Thank you.
Okay, so next up we have Patricia Turner.
Great. Thank you so much. And today I'm going to be talking about implementing sustainable welfare practices in research settings, adoption of non-aversive handling of mice. And this will be a little bit different from some of the other talks. We've had some fantastic research talks so far. This is really going to be focused on translating and applying research in a real world setting. So I'll provide a few definitions for people not familiar with aspects of non-aversive handling of mice, a little bit of information about what is known about mouse preferences for handling and for rats as well. The approach for our project, areas of emphasis for change management, and then kind of summarize future steps for this. Okay, so what we know about about rodent handling is that really through almost 16 years of research now, non-aversive or low stress handling really refers to how mice and rats are removed from their primary enclosure. We know that tail handling, particularly for mice, is aversive, and they don't really habituate to this method of handling, even if it's done really carefully by tender-hearted people and in working in vivaria. And it has negative effects on health and other behavior outcomes. So low stress handling methods then, we talk about tunnel and cup handling. These are considered non-aversive and again have been shown through various studies that they are so. They are thought to be better for health and behavior problems. They promote consistent and quality study data, and it reduces risks for biting for the people handling them and for injury for the animals as well. Takes as little as 2 seconds at each cage change to start habituating animals to tunnel or cup handling. And tail handling or scruffing of the animals, once you have them outside of their primary enclosure, doesn't counteract the positive effects of the low stress handling when removing animals from their home cage. So what does this look like? So this shows sort of the traditional handling methods that can be used. And I'm talking today about handling methods in use at Charles River. And these are the methods approved at most of the facility SOPs. So tail handling here on the left, that's a traditional method of lifting animals from the base of the tail with fingers or forceps. In the middle, this is tunnel handling, and that's the process of guiding animals into a tunnel or a similar device. It could be other type of cage furnishing to remove them. And then the last one is the cupping, or cupping with 1 or 2 hands of the animals to scoop them out of the cage then and put them onto another surface or into another cage. What's the background on this? I don't really have time in this short talk to go into this extensively, but there have been many papers that have demonstrated the importance of tail handling on physiology for mice. So you can see in this panel here on the left, if we even look at breeding, if we look at animals that have been tunnel handled or tail lifted, we can see significant differences in numbers of pups weaned per pair and born per year. And then when we look at physiologic data over here, when we use cup handling of mice, that improves glucose tolerance in these animals, reduces corticosterone levels, and we can see even reduced size of adrenal glands in these animals. So overall, we can see improved breeding outcomes, larger pups, more pups born and weaned, longer reproductive lifespan. And it's thought that these then contribute to improved research models and data quality as well. Oops. A little more data here too. Again, when we look at even things like reducing urination and defecation with when animals are handled here on the left, using tail, tunnel, or cup, again, reduce urination, signs of stress and defecation during handling. And then many, many studies demonstrating, and I'm providing some references here on across labs, across strains, and across different types of tests as well, that this improves, reduces stress and improves overall well-being whoops, oh my gosh, sorry about that, improves this over the lifespan of these animals. So taking all of this evidence in mind then, we decided then that, we would like to take this on as a challenge. And I do want to note that while there's extensive evidence for mice, the information about rats is missing. There's not as much evidence. There's only a couple of papers out for rat preferences for handling. And this was a bit of a risk for us when we were deciding to launch the project, because we had less evidence to present, and we knew we were making some assumptions for this as well. So our problem really, when starting out with this, was my group, Global Animal Welfare and Training, was a small group of, it's a small group of 11 people. So it's a corporate group. Can't demand change. The only way that this group can get anything done is through consensus building. And as indicated on here, we're a national contract research and rodent breeder, operating in 21 countries, so 110 unique sites working with animals, 9 languages, 10,000 employees working in some way related to animals, study directors, veterinarians, pathologists, technical staff, caregivers, managers, all of these other people with different laws, standards, traditions, and cultures related to mice and rats across all of these different places. In addition, there had been a well-intended but misinformed global training by our environmental health and safety group, who were very concerned about the number of rodent bites that employees had been reporting as well. And so they were very keen to ensure that people were not putting their hands any closer to rodents when they were working with them. They really wanted them to be as far away from their mouths as possible. And the other thing is that it's, again, not a consistent business, in the sense that it's multiple major business units that are held together with separate leadership. So animal breeding and research models, a large part is safety assessment, but also discovery services, biologics, microbial solutions, Cradle, which are like separate little research facilities in which outside investigators come in and do their work, and then insourcing solutions where employees go into other research facilities and they conduct work there. So it's trying to think about how to put together this change management that will address all of these different settings. So a little bit of a challenge, when we're thinking about this. I did want to step back. I'm not going to talk about this extensively, but I did want to step back and say that alongside of this, we had been working as an overarching strategy explicitly to enhance and improve culture of care at Charles River. And this is part of ensuring that there is a fertile ground for making animal welfare changes. If we had tried to start this project 2 years previously, we likely would not have been successful. And just for those unfamiliar with culture of care as it applies to caring for and working with research animals, it really emphasizes institutional responsibility for our ongoing and sustained process to ensure good animal care and welfare, good well-being of those caring for and working with research animals, excellence in research quality and data reproducibility, and enhanced openness and societal transparency. Really about exceeding a culture of compliance, and this emphasis on care, respect, and responsibility is important. And of course, it's aspirational and evolving. You're never done when you're thinking of enhancing your culture. So low stress handling was selected as one of the projects to go forward with and to prioritize because of the millions of animals at Charles River that it could impact across the organization. And so that comes into this culture of care aspect. And again, I'm not going to address for this group in too much detail aspects of total quality management, but on the left-hand side here, many people will be familiar with some of these principles here of total quality management, which emphasizes employee ownership of issues, basing the work on process and developing systems to integrate the change you want to make, communicating this, driving it through data, trying to have constant improvements, and always focusing on the customer, which could be the sponsor, could be the patient, thinking about those sorts of things. And it's a continuous improvement activity. And again, this part on the right is in part where you're encouraging continuous incremental improvements to make your goal. You can also achieve change through using radical change. Examples of those might be a new platform, a really new method, a new strategy, a new philosophy, such as smartphones, personal AI assistants, the internet. Those are all radical changes. And those are really important to push for innovation, but it involves a lot of risk. They are really resource intensive, and it can be very disruptive, especially for a business. And they're much rarer events. When we use incremental innovation, that's the process of making these small, gradual improvements or advances in our techniques or services, that those can then be accepted. And you then have this culture where you're sort of making these incremental and small steps, and that really helps to sustain improvements in the long run. And you need both, of course. In the long run, you need some of both, but incremental improvements is something that's more likely to garner executive approval when you're trying to implement change. So for this particular low stress handling project, what we started with really is that tail handling of mice is ubiquitous in North America and elsewhere. So a really big challenge to try to implement this and make this change. It was really helpful for us in that at the beginning, we had a few clients and sponsors that were just starting to ask questions about handling practices at Charles River. To really launch this, we needed to develop clear, simple messaging for the campaign. So, tail handling of mice and rats is aversive, period. And then tie it back to business, scientific quality, and animal welfare. We also needed to address the environmental health and safety concerns about rodent bites, because of course, safety of employees is really important too. And the first step was gaining leadership support for the project. So a lot of communication at a lot of levels. We developed a 7-minute e-learning module for senior leaders and asked them to cascade it to their teams. And then, when you think of what you're asking, it's asking all the operations people, the managers, supervisors, caregivers, and technical people to layer this training and change on top of everything else that they're doing in their day. So it's a really big ask, and no matter how passionate we feel about these sorts of changes or how right we think they are, it's a huge imposition, particularly for more experienced employees who are really going to have to stop and rethink and question everything they do. We also worked with our internal 3Rs advisory group to help prioritize low stress handling as a company project, and that helped lead to the launch overall. So really busy slide here, but really this is the project map for us when we're looking at this project. I talked about the training of the senior leadership, the 7-minute e-training, and the benchmarking survey that we did at the very beginning of the project to see where we were across all of our facilities, a clear objective that everybody could wrap their heads around. Looking at rats, we conducted a survey looking across all age groups and life stages at all types of businesses. We found that for 80%, for rats, 80% of our sites were using tail handling for at least some of the groups, and 100% were using it for mice. And so we had lots of work to do. To get this going, then we had looked across our business groups, developed training, and got the buy-in. Had the sites begin testing this, so pick their champions and doing little pilot studies with this. We had constant messaging through newsletters and other things, little quizzes and games for employees with prizes and things, listened to the challenges, took feedback from them, tried to connect different groups together. We did not have all the answers for this. So it was about connecting people who had found the next step to try to make these things work, and then providing training or developing new training to help them, monitoring progress and doing this continuous feedback loop. That's the quality improvement process, and then recognizing and rewarding sites. So I'm not going to go into sort of explaining all of these other things, but we looked for collaborators, vet champions, other training people who could help us with these, and did a mid-year, sort of a midpoint assessment and a survey of where we were with this. Just to emphasize, education and training is really the backbone whenever you're trying to do change management. So we developed an internal low stress handling resource page that had training materials for all different levels, all different businesses, with videos, webinars, presentations, a lot of external resources, including even vendors and things where they could get their tunnels from. We kept all of the training really short and to the point, and provided a lot of transparency and feedback of surveys to people. We also needed to set clear KPIs, key performance indicators, to ensure that sites knew when they had achieved the goal. This just wasn't an endless, forever project sort of thing. We wanted sites to know. We set an 85% target for mice and rats at the site, and and showed them how to do it through sort of messaging and PowerPoints and things that we sent out. We told sites to expect failures. We had a plan for how to deal with them. And we also tried to prepare supervisors for recidivism and train them to avoid punishing employees, because change is really hard. So just to summarize then, really the culture of care piece we thought was really important for even being able to start this project from sort of a care-based point of view, respecting the animals. We recognize this to be a continuous improvement process and not a stopping point. Major shifts in animal welfare, such as changes in handling practices, require thoughtful approaches to address concerns, doubts, and challenges, and you really need to be flexible and develop appropriate education and training resources and set realistic KPIs for it. And building engagement may require the thought leaders, the people who may have started it, to step back and let the local teams step forward to really develop competence and ownership for true sustainability. So thank you so much for your attention, and a few readings as well for those interested. And I just mentioned too, sorry, I forgot to mention, one of the things that we kept on our webpage too was the target. So this is kind of the thermometer of progress. This was as of the end of May. I retired from Charles River then, but there were at least another 10 sites that were incrementally away from this as well. So that is in front of people all the time. They can see the progress as well in there. Thank you.
Thank you very much. My question has to do with the way that this sort of progresses within the system, because the idea that mouse handling using tunnels is less stressful has been around since 2010. And it must have reached, I imagine, a number of people in different places in the organization fairly much earlier than when you initiated this general program. I don't know if you could say something about that.
Yes. So the timing, that's sort of what I was saying. The timing had to be right. So people have been obviously talking about it, thinking about it. We had some sites that were already almost there, that were really true champions. So it was asynchronous, and it's still asynchronous, and that's okay. Sites have different resources, availability of personnel to work on some of these things, and you have to trying to force everybody to do everything at the same time doesn't work. But you have to reach a threshold, which I think is what you're almost implying there, right? That threshold at which enough people hear about this, there's buy-in, you start having clients asking about it, And it's like, yes, we can finally go ahead with this. So it all really came down to trying to pick the right time. We wanted to start it for some time, but you know, did a soft launch and a soft rollout first and had a number of external speakers coming in, doing company-wide seminars on the topic as well to kind of lay the ground for it too. Great, thank you.
Okay, so we're switching gears to the shorter talks. The fourth one is not going to happen because the author couldn't be here. So we'll have 3 short talks and then time for questions for everyone. So starting with Anissa.
Hello, everyone. I'm going to give you a brief presentation about some results of my PhD, which focus on the analysis of the effects of temporary outdoor access during the fattening phase of the pig welfare, health, and performance. Indeed, most of the pigs are raised in a conventional barns inside, and it appears that an outdoor access can potentially improve the welfare of the pigs. For that, I follow 300 pigs in 3 batches, male and female, from day 76 to day 160. Half of the pigs were raised in a conventional barns without any outdoor access and were called O-, and the other pairs were called O+, were raised in conventional barns with an access to a pasture twice a week for 4 hours in the morning. To characterize the behavior of each group inside and outside, I analyzed the time budget of the pigs at the beginning, middle, and the end of the experiments. In this table represents the average time budget of the pigs of these 3 periods. In the morning, when the O+ were outside, they spent less time resting and more time exploring the environment when the O- were inside. In the afternoon, when all the individuals were inside, O+ were more rested after going outside. Furthermore, at the end of the fattening phase, at 150, we show with a body injury scale that enable us to highlight that O+ pigs showed significantly less severe body injuries than O-. Then this graph represents the median level of severic cortisol on day 104 and 150. And we show with pigs O+ were in green and O- pigs in brown, that O pigs have significantly lower salivary cortisol than O-. Finally, both of groups show the similar growth performance. So this study showed that temporary outdoor access can have positive impacts on the welfare and the health, but no effect on the growth performance. So our results should provide a better knowledge about the consequences of giving an outdoor to access to the pigs. And you need more details, you can find these details on my poster. Okay, thank you very much.
Next up, we have Jennifer.
Hi, I'm Jen. Thanks for the opportunity to present my poster about the influence of education on positive reinforcement training with goats. So when it comes to the 3R, refinement can be implemented as with positive reinforcement training. And I think you all agree with me that cooperation is better than restraining, since restraining is always stressful for the animal. But if you want to implement cooperation, you need someone who trains the animals, so an animal trainer. And unfortunately, there's a lack of animal trainer research, and that leads to a questionable comparability when it comes to animal training as a refinement measure. So we ask us the question, how to become a good and successful animal trainer. And as Bob Bailey, a well-known animal trainer, said, training is simple but not easy. I think that fits very good to this question. It's a very simple question, but not as easy to answer. But we tried. So we had two educational programs. We had 15 volunteers in a workshop group. They got a 2-day workshop by an experienced animal trainer, and a self-study group with a book about animal training or positive reinforcement training in particular. So both groups, every participant was matched with a goat. They had 2 weeks time to train the goat with one training goal. And the training goal was the following. As you can see here, the goat had to jump on a platform and had to lay its chin on the trainer's hand and tolerate a touch of the neck by a second person. On the left here, you can see Viviane Tebi. She's the experienced animal trainer who taught the workshop and was our reference standard. So we did a video analysis with a developed protocol after the 2 weeks of the training sessions and found the following results. So after all, the training group was better in the training. They received higher objective training success and therefore better grades. And as you can see here, the light blue scale is a workshop group, and only participants of the workshop group received or succeeded completely with the training goal, and only in 3 cases. And interestingly, the training duration was in both groups quite long, so about 2 hours of training with not perfect success. So they didn't manage to train the goal completely in those 2 hours and referred to the reference standard, who needed about 30 minutes to train. It was much longer. And that led us to the conclusion that compared to self-education with a book, a 2-day workshop still does not make perfect animal trainers, but helps participants to train more successfully. And if you want to know what that means for the practice and what I hide in this back here, I'm very happy to see you at my poster. Thank you.
Yeah, thank you very much. Good morning, everyone. I'm just here to talk about a study on the impacts of a prison-based dog training program on the behavior and welfare of the animals. So to start, what are these prison-based dog training programs? They are basically programs where dogs are taken into prisons to be trained by the inmates. And what do we know about them? We know that they have been on the rise worldwide, and that research surrounding them has mainly focused on the impact on the human participants, namely on the inmates, while little information is available on the effects on the dog counterpart of the equation. And thus, the aim of this study was to assess the impact of such a program on dog behavioral skills and welfare. And we did this in collaboration with a recently started program in Portugal called Pelos Dois, in which shelter dogs are taken to prisons here in the north of Portugal to be trained by the inmates. We collected data from 45 dogs allocated to 3 different prisons, and we evaluated both dog behavior and welfare indicators before, during, and after the program. And very briefly, regarding dog behavior, we assessed, for example, how these animals reacted to being handled by a human or how they behaved around other dogs. And we found that, for example, among the categories that we analyzed, that these dogs with the program, they improved their sociability towards humans. Relatedly, we also assessed basic obedience skills, such as obeying to commands or walking on a loose leash. And we found with the program improvements for pretty much all of the analyzed behavioral skills. Regarding welfare, we collected saliva samples from the animals at 3 time points during the program, and we collected samples at the shelter to compare with those collected at the prisons, and we found no differences overall in cortisol concentrations. Finally, we also exposed the animals to cognitive bias tasks to assess their affective states. And also here, we found no differences in the dog behavior from before to after participating in the program. So in conclusion, the participation of these animals in the prison-based dog training program improved their behavioral skills and had no impact on their welfare. And this data suggests that this type of programs, besides being beneficial for human participants, which we know from research that is the case, that they also may be beneficial for the participating animals. Thank you very much for your attention, and please check our poster for more details and information.
Just a question for Jennifer about the goats. So there's clearly like a really good, difference between like hands-on training and book training. But say you're like learning a new protocol from literature or something. Is there can you suggest any compromise for book training that might still have hands-on supervision?
Sorry, could you repeat the question?
Is there any kind of compromise you can suggest where if you can't have hands-on supervision, you're kind of forced to use literature or online resources?
Yeah.
You could suggest.
I could think that the book would be a great basis for the theoretical parts of animal training. So during the workshop, they also had a theoretical part with theory on operant conditioning and something like that. And I think it would be very nice to have that as a book or an online course, for example. But in the end, it really needs practice. But you can do the practice at home or at your home facility as well. So yeah, I think there could be a good combination, but it takes time both.
Yeah, thank you, Beate Nielsen, UFAW. This is for Anissa. You showed some cortisol results, and cortisol is a measure of arousal. And I was just wondering if you were surprised by the difference you found, because you found that the pigs that were not outside had higher cortisol than the ones that were. Could we not expect pigs that were outside moving about to have been more aroused and therefore have a higher cortisol level?
I'm not surprising about this result because we have a lot of data with the behaviors, the health, and we see that that O+ pigs with outside the access are less resting, and it's corresponding to the arousal because the salivary cortisol are lower for these pigs. And we have similarly in result with the hair cortisol, with its chronic cortisol accumulated in the pills. So we're not surprised. We are satisfied with this parameter.
Hi, Steve Wickens, UFAW. So it's a question for Anna. So you've done this you've done this work. Clearly, it seems to be good for the dogs, good for the prisoners. So I presume at the end of this, the dogs are then put up for rehoming. Did you go further and then look to see whether or not the dogs that had undergone the prisoner training were more likely to be rehomed and then stay in the home than dogs that were in the shelter that hadn't gone through that?
Okay. Thank you, Stephen. And a very interesting question. And actually, we have that as a goal for the future. We couldn't do this this was the first time we actually worked within this program. But our aim for the future is if they continue with the program, if this program continues, that we really want to check if these dogs are more likely to be adopted and how well they adapt to their homes as compared to dogs that do not go throughout the program.